It's actually meant for students to do general general experiment, mixing solutions, and also prepare their reagents for maybe a more specialized experiment. This is the genomics room. This is the place where most of the students do the extractions of the genetic material from the samples they receive, one for the DNA, one for the RNA. Alright, and we have some machines around here that we use to determine the quality of the genetic material that they extracted from the sample.
Hey now, come on, let's go. The music's playing, I've got to move on.
The young electrophoresis room is the place where they do the analysis after they have extracted the genetic material. What they do is they want to do analysis. Basically, want to check whether there are mutations, whether or not if anything's gone wrong with the genetic material. Obviously, they have to do something to the genetic material first that they have, whether or not if they want to amplify a specific gene they want to study, before they actually run it through agarose gel like here, the one which is going on over here. So this is like agarose gel made of agar-agar. It's like agar-agar. So what they do, they make a hole at the agar-agar and then they put the genetic material and let it migrate from one end to the other end of the agar. And the agar is actually served as a medium, and because they build up resistance for the genetic material, the smaller piece of the genetic material will move faster because they have less resistance compared to the larger piece. So from there you'll be able to build out a genetic profile. So after they have done that, they will do some staining, they put some color to it, then they will build it in the gel box here, G box here. PCR stands for, I think most of the students have already learned that, PCR stands for polymerase chain reaction. It's a method used to amplify a very small amount of DNA. For example, when you receive certain samples from crime scene, so you get only very small amount of sample, and the genetic materials obtained from the extraction is going to be very little. It won't be enough for us to study our gene of interest. In order to study the gene of interest or a particular regions of our genetic material, we will then amplify them. So this is a place where we prepare all the cocktail. We call it cocktail. You mix a lot of reagents together so that before you can amplify them. So we prepare all this cocktail that I mentioned in a small tube like this. In a small tube like this. So you put all your ingredients, like how you make your cake or make your whatever syrup drinks, right? So you put in all the ingredients in this small tube using all those pipette. So once you have prepared everything, so you cover it, and because you have all the ingredients all over the tube, right? So what we do is we put into the spinner, alright, and close it and turn it on to spin them. Alright, after a while you'll find that you'll be able to accumulate all your reagents and solutions at the bottom of the tube. As I said, this is a PCR preparation room, so we're going to put this tube into machines because we do not, it involves many different steps, so the machines will do the multiple steps for us. These are the PCR machines, just like your oven now. So you have now make all the ingredients for your cake. We make our cake in this small tube. So after that, you put your cake, whatever dough, into the oven. But what we do is we put this tube. So remember, we want to amplify a specific segment of DNA or genetic materials that we have obtained. So we put the tube into the machine. Alright, and then we do all the settings that we need. Alright, for example, again we have to set the temperature, how long we want to keep the sample in that temperature before we move on to the next temperature, and so on and so forth. And obviously the whole amplification process, the PCR process, involves heating process, cooling process, and also like baking process. We keep it at, let's say, 60 degrees for a certain period of time. So once you have put the sample, turn it around, on. And after 2 or 3 hours, it's all done. You can remove the tube. Alright? So you have all your targeted genes or whatever material that you want to study amplified. to billions fold. So from one copy, let's say one copy of that genetic material, you will now have maybe billions of them. So you can now see this under your in the agar gel that we mentioned just now, the agar gel. So we just take some of this and put into the agar gel. After we run it, so we can then put some color to the agar, and we can see it under the machine.
Hey now, come on, let's go. The music's
So what we have is a bacteriology room, a place where we actually grow bacteria. Okay. In many instances that we use bacteria as a host to help us to grow certain genetic material again. So let's say from the previous sections that after you amplify your genetic material, alright, and you want to even get more out of it, or you want to manipulate, for example, you want the genetic material to produce it. As we know in the field of genetics, you have genetic material that eventually will give out the protein in your cell. So the protein is the final functional unit, okay, that make up who we are. So now if you want to make use of the genetic material that we have amplified, so that we can instruct the genetic material, for example, to produce the protein. So we can do that by making a clone. So the clone, basically, in order to make the clone, you need to make a lot of them before you can make the protein. So for that, we actually make use of the bacteria. We get the bacteria. So the bacteria that we have here is actually E. coli. I think most of the students already learned E. coli, Escherichia coli is one of the gut microbe. Okay, and most of these E. coli are actually engineered, and they do not infect human. So we use them to help us to grow the clone that we mentioned, so that you can use it for whatever downstream applications later. After we make use of the bacteria to produce a lot of this clone for us, we can inject the clone to the animal, to the certain part of the animal, and then that will allow the clone, okay, to direct the production of protein in particular regions in the animal that you want to study. So having the protein in that region, it means that you actually study what is the role of the protein, right? So you have a lot of this protein now in this organ, for example. So if it's playing a role in improving the health of the organ against the infection, then animal with this clone in their body will resist to, for example, certain infection when you give them, the bacteria or the pathogen that cause the infection. So this is how we study what's the role of this genetic material that are translated into a protein that eventually that you can see in our animal experiments.
Hey now, come on, let's go. The music's playing.
This is one of our animal surgical area. So we do actually do surgery to animal. The machines that you have here basically put animal to sleep. That's like how you put animal to, just like you put human to sleep. So you put the animal in here, put them to sleep, and later on we will then do all the surgery somewhere here. Okay, so throughout the surgery they will be anesthetized as well, so they won't actually wake up. So what we do, you can cut open whatever and then do whatever you want. After that, you put everything and suture everything back. So what I do is I cut open pregnant mouse with their baby in there. So I take out their baby still within in the placenta. So I inject the so-called clone. Remember I mentioned about the clone? So I inject the clone into the brain, and I want to see how is this particular genetic material will affect brain development. So I mentioned that when you have the genetic material, you can get the genetic material to produce the protein. So you can study what is the role of the protein or what is the role of the genetic material during brain development. So what I do is I have this mouse, pregnant mouse. We cut open the abdominal area and then remove the embryo. Still attached to the placenta, they are still living. So throughout the whole surgical, we inject the clone to the brain, and after that we use electric to give them an electrical shock at the brain. So when you give an electrical shock, that the cell will take up the clone. So the clone becomes a part of the cell, just like how the cells having the original genetic material, now they have extra genetic material that we introduce. So after that, what we do is we put all this embryo or this baby back to the mother's mother's tummy, and then we suture it. After suture, we apply all the analgesic painkillers. So after that, we allow the mother to recover. So throughout the whole surgery, it takes about 1 hour, and then once mother recover, the mother will still have the baby. They will still continue bearing the baby until, for example, until birth. So after birth, you can actually take their baby and then study what's wrong with the brain after we introduce the clone into the brain, as compared to the baby without the clone itself. So you can compare. So when you have this clone, it produces this protein. It makes the brain develop in a different way. So then you can tell what is the role of this genetic material or the role of the protein during brain development, as compared to the untreated mouse embryo.
Hey now, come on, let's go. The music's
This room over here is actually animal husbandry area. So this is the place where we keep the animal, and when we are not doing anything to them. So this is the general husbandry, and this is basically where they live. You'll see that the animal will have the bedding to absorb all the waste produced by the animal, and also they will have unlimited access to food pellet as well as water. Alright, the air going into the cage are all filtered, so they are pathogen free, and the air coming out of the cage is also filtered, so the user or the people in this room will also be safe. So none of the infectious agent can get into the cage, and none of the infectious agent from the cage can get outside the cage as well.
Hey now, come on, let's go. The music's playing.
This is place where we culture cell, mainly cell. So it can be any cell. It can be cell from patient. When we obtain, for example, cell from breast cancer patient. So you can culture the cancer cell that we remove from the patients into culture dish. So the idea of doing this is to get as many cells as possible, so that it can grow on a dish like this. Once it's on the dish like this, you can do a lot of manipulation. For example, testing a drug. So instead of testing the drug before, directly on the patients with breast cancer, you can now test the drug on the breast cancer cell that you culture from patient. So if the cell, at least if you find out the cell is actually dying, then you know that the drug might be effective. But having said that the drug might be effective in killing the cancer cell, doesn't mean that the drug is actually not killing the normal cell. So a good drug is only targeting at the cells of interest. For example, a good drug for breast cancer will be a drug that kills cancer cell but not the normal cell. So that is how we can actually do various screening using cell, or in fact cell line. Okay, so cell line, there's a few type of different cells. So primary cell is the one that I mentioned just now, like a cell that we establish from patient. Alright, but there are also cell line that also established from patient, and it becomes immortalized. It means that the cells will not die. So every time that you need to use the cell for any kind of screening, you can always culture them again and again. And we keep some of these cells in liquid nitrogen tank, like that, which give you a temperature of below about minus 180. So what you get is the cells will become immortalized, and they will stay in there for a while. And obviously the cell won't get immortalized straight away. It means that you have to do something to make it immortalized, and eventually you keep them in the liquid nitrogen for long-term storage. So perhaps a few years later, when you want to study the cell again, you can remove the cell and then regrow them on the dish, on the tissue culture flask like this. Alright. In order to make them immortalized, you have to give them certain again genetic material. For example, you see cancer cell grows so fast, and becomes uncontrolled because they continue to grow. Alright. When they continue to grow, they continue to spread, and they will settle at a new place, and they start to grow there as a tumor again. So the reasons they become cancerous, obviously because some of the genetic materials becomes defected. For example, mutations, caused by various reasons, including radiation, for example. So because of the mutations, these cells become defected, and they keep on growing. So cells like that, right? For example, the cell line that I mentioned, they can grow, continue growing as long as you supply them with the medium. So we have a lot of enriched medium that we use to culture, used for culturing different type of cells. If the cells from breast cancer or a cell from, let's say, brain, require different nutrient. So where do we grow the cell? So you have to make the cake, you have oven, right? So we have our incubator, something function like oven. Okay, we give them the best temperature, 37 degrees Celsius, and also the best condition, carbon dioxide condition that mimic the inner body of a living organism. Alright, so our temperature is about 37 degrees Celsius. Alright, that's how we try to mimic the inner body of a living organism, so that the cells will grow happily in there.
Hey now, come on, let's go. The music's playing.
So we call this dark room because we can do a lot of, it's actually meant for microscopic imaging purpose. This is actually an fluorescent microscope. So remember I mentioned about the clone, for example, right, that produce protein that we put into, let's say, a living organism, and then after that we look at what is the outcome of that. Besides looking at only at the morphology changes, we can take all this tissue and then make into a section, okay, just like a histology section, and with the sections you can even stain the protein with a fluorescent marker. Remember the marker I mentioned about? So the fluorescent marker, so if the protein is there, then perhaps the sections become fluorescent green or fluorescent red. So what we can do is we can use a microscope, fluorescent microscope, to look at those sections and check which part of the brain, for example, is actually having green color fluorescent. What does it mean? When you have green color fluorescent, it signifies, for example, the protein is there, right? So whenever there's no green color fluorescence, means the protein is not there. So you can actually zoom into a microscopic level, okay, to check what exactly the protein actually does to the brain that leads to such morphology, you know, at our course examination. So from something that you are zooming all the way to a single cell level. So the same thing, the class that we have there, okay, if we culture it, we have a different microscope upstairs, which is inverted fluorescent microscope. So you can also detect certain protein by label them with certain fluorescence marker, so that you can then see them under the microscope, even directly from the flask itself when they are still living. So those are what we can do. So basically these are all the different equipment that we use to analyze the behavior and the outcome of certain treatment that we perform onto the cell or onto the living